So good morning, everyone. It's fantastic to be invited to speak here, and lovely talking to you all this week and hearing some fascinating talks. And what I want to chat about today is some work I'm doing with colleagues in Oxford mainly, and it's a work in progress. It's really kind of a methods talk with some applications. And what we're trying to do is use an approach that, in principle, should be a really good idea, which is to try and study, the history of humans, in particular our size changes, population splits, and hopefully in future things like migration events, by building coalescent trees for samples of individuals. So it sounds like a really good idea, and I'm going to talk about, why it's difficult and a bit of progress we've made. Okay, so why does it sound like a good idea? I think this audience probably well aware of what's on this slide. So this is, of course, a coalescent tree. Up in the top right-hand corner. And the idea is something we know. If we take a sample of people from this room or a sample more generally, we know that at each particular place in the genome, our history can be described as a coalescent tree. It might depend on the populations we use and so on. Certainly will. And as we move along the genome, we know that those trees will change. And in population genetics, almost all the models that we use, no matter the question that we're interested in, maybe we're interested in understanding selection, or in this talk, population history, no matter almost what we're asking about, if we knew those trees, they would sort of fully capture the information about the underlying process. Okay, so if we could learn these coalescent trees, we could sort of use full information approaches that capture all the things that we could possibly learn from diversity data. And the only problem that we have is that, of course, we don't see these trees. What we see is patterns and variations, and we have to learn the trees from those patterns. So in principle, we capture sort of the information. I sort of want in this talk to give you kind of personal sense. I also have the idea of trees being, in some sense, a good thing, in the sense that they're constrained. So if we can take data and build trees from them, then we're kind of forced to put all the variation data that we see, on this object. And so in some sense, they're nicely internally consistent. And in the future, one might hope that that could help in checking things like modeling assumptions. There was some discussion of that yesterday that was fun. And also kind of critically evaluating our models and updating and improving them. So they seem like they should be a good thing. On the other hand, we've heard lots of really great talks over the last few days about understanding human population history, and most of them, at least, haven't directly tried to build these trees, and they're not generally widely used in the field yet. And the reason for that is that it's a very, very difficult problem to kind of go from this variation data. This is a set of, how many are there? 6 sequences and 8 sites, or so 1, 2, 3, 4, 5, 6, 7 sites even. To go from this to the underlying tree is a very challenging problem, and it has no exact analytical solution, even if you think that our history in the past is the boring old standard coalescent with a constant population size. Even in that case, it's hard to go from here to here. On the other hand, people have been very successful in developing highly sophisticated approaches, exemplified maybe by the software Beast, that can do this for a single locus or a small number of loci even, like the mitochondrial DNA or the Y chromosome, and they have a special property, of course, that they don't recombine. Okay, so in that setting, things like MCMC can be used to sample from the trees given the data, but the problem with them from our point of view for this talk is that these methods are very, very computationally intensive and a bit too slow, and that's because if we're going to say things about human history, for example, because our mutation rate is rather low, we don't get that much information from a single piece of a genome, and because trees are highly stochastic, so we need to combine information across, I'll argue, hundreds or thousands of loci, and it will help if we can analyze large sample sizes, and these methods can't do that for us. And in order to get these large numbers of loci with lots of different trees, we need to study autosomes, and there's a really big problem of studying autosomes from this point of view, which is, it's sort of been kind of an interesting thing in my career to study, but the problem for the point of view of this talk is recombination. So recombination happens and makes this problem even more difficult, although methods have been attempted. Okay, so what I want to talk about today is, as I said, it's a work in progress, but the basic idea of what we're doing is to try and construct trees in many parts of the genome that we can think of as almost non-recombining. So we sidestep the recombination problem. And so the conceptual idea, I like to think of it as sort of finding lots of autosomal Y chromosomes and mitochondrial DNA segments scattered around the genome, and then we can try and build trees for those and combine information across them. Okay, and I thought I sort of switched the order of the slides around a bit sort of to try and convince you early that this wasn't a completely crazy idea, that we could try and find such things in the human genome. And what we did, we started this project by looking for regions of the genome at least 20,000 bases long that had sort of little or no evidence of recombination in the following sense. They had to have a rate which is less than about a 20th of the average in the genome over that whole 20 kb region in each of 4 completely differently produced genetic maps. And these maps are made they sort of span everything from current day pedigrees, that's the decode map, so that's people today, African Americans, so that's sort of people from Africa or African and European ancestry in the last 6 generations, and LD-based maps, which of course are in the deep ancestral past. All of them have to agree in multiple different populations that there isn't much recombination. So hopefully it's a strict condition. And we found quite a lot of regions like this because of the hotspotty nature of the genome, averaging 35 kb in size, and totaling sort of 4 or 5% of the entire genome. Okay, so it is a sort of relatively small fraction of the genome, but it is about a chromosome's worth of data. And I think I might argue that hopefully we can increase this maybe to 10 or 20% without too much difficulty in the future. And to give you a sense of how much data we get from this, we get stuff across the chromosomes. On average, each of these regions has about 190 sites in 1,000 genomes. And so we can think of these trees for these regions, if you think trees are appropriate, as being pretty well resolved. We're going to have quite rich information about the relationships of individuals from these trees, and they're about, a chip's worth of data, if you like, in total.
So
okay.
How does that compare to the density of mitochondrial SNPs on the mitochondrial tree?
I hope someone, could anyone else answer that question? I don't know offhand.
For a sample.
I mean, it's tiny compared to Y chromosome, right?
It's tiny compared to the Y chromosome.
Yeah.
Like, I think is the reconstruction going to be better or worse than the mitochondrial tree?
I think there are thousands of available SNPs in maybe it's. Yeah, I think it's somewhat less than mitochondrial DNA, but I'm going to not declare a definitive answer. I mean, I think what the hope is we're going to win by having many independent runs of history. Okay, so we may not be quite as well resolved in each one, but we're going to have many independent runs of history.
You're preaching to the choir.
Good. Okay, so we've the data we've applied to so far is from 1,000 genomes mainly. I'll talk a little bit about Neanderthals at the end, and we've analysed up to 1,000. We've analysed 1,000 haplotypes in total, but at the moment our method can only account for pairs of populations, and so we're sort of averaging up. We're analysing up to hundreds of individuals on these trees. And there's an important point that holds throughout this talk. We assume the infinite sites model, so we're imagining that the mutation rate is low enough for that to hold. It's probably true for humans reasonably. And in some cases, due to either a little bit of recombination in these regions or perhaps errors in the 1,000 genomes data, we did find the data weren't compatible with infinite sites, so we sort of removed the minimal number of sites needed so that we got for each region data compatible with a tree. Okay? So that's how we started, and we accounted for this by normalizing the mutation rate in the subsequent analysis. Okay, so the hope is there's quite a bit of data here, and I'm going to try and convince you that the results and the trees we get from applying to these data are consistent with stuff that we know. Anybody? Graham?
So you say you remove 30 regions with recombination, but then you're also removing sites which fail the 4 gamete test?
So there's some rule, which I can't quite remember, that said, if in a single region you remove an appreciably large amount of the sites, then you chuck the whole region away. Because what you imagine there is that's almost certainly a real LD breakdown. So even though you've got this condition, there can be some small amount of recombination in the regions. So one important thing that you might ask me to do in a future slide is to check robustness to some amount of recombination in the regions. Yeah, Molly?
So these regions end up really far from genes?
No, I think they're more likely to be slightly enriched for genic regions than anything else because they're required to have low recombination rates, and on average recombination rates are lower in genes. We haven't surveyed it in detail, but it's an interesting thing to check. Yeah, Andy?
Graham's question was about gene conversion. So if there are individual sites that have,
Graham's denying it.
organic
But it's a good question. Well, as far as we know, I mean, we don't know everything about gene conversion for sure. As far as we know, it's mostly seen in hotspots, so it's usually accompanied by some crossover information. So we don't know of any, I think, gene conversion specific hotspots in humans, for example. Now, if there are very localized gene conversion hotspots, we should take out the sites. And they wouldn't affect the wider region. That's how gene conversion works. So I think it's okay. I mean, there are some issues with doing this in that we're better. We don't always see incompatibilities, even if there's been recombination, right? So it isn't perfect, and it's an approximation.
Well, what's the largest of your regions?
I don't know. I think it's like 100 kb or so, but.
And are these regions a bit what you would have expected just by chance, or do they have something special?
Are they, in what sense? Do you mean that?
If you assume some kind of regression rate, uniform version of the genome, so maybe what would be the factors that would show no recombination?
I think this is completely consistent with a hotspotty genome. We wouldn't, if we had any sort of flattish rate, we wouldn't find many of these, but essentially the model of large LD blocks in humans is an approximation, but for some of the genome it holds pretty well. Okay. I think the proof of the pudding might be in the eating in some sense, so we can sort of look at how it works in a minute.
So just following your original motivation, I'm not familiar with it, but I wonder, is there any previous information on sort of very simple models of demographic structure in which the kind of ascertainment that you're doing here leads somehow gives a different answer than any kind of biased answer? In other words, if we sort out those regions that happen to not have any recombination, do we know whether they give any substantially different results for structural questions from a theoretical standpoint?
So we try to we worry that they might have a different mutation rate to other parts of the genome, so we try to normalize that away by, so what we do, and it's sort of under the hood, of course, but it's sort of, and it's also tied up with this. We needed ancestral site information, for example, to do this test and to sort of get the root relative to the data. So that removes some sites because we didn't have chimp information. And then we have some removal from this. So what we did is we kind of normalized our mutation rate estimate to account for this, but it would also account for whether these regions have a lower mutation rate on average.
But your estimates of mutation rate are based on within population or between population?
Within
or
populations,
between
yeah.
Huh?
Within populations.
Oh, okay.
So we felt that was somewhat
the
better.
site frequencies, basically, or not?
We used pairwise diversity,
Oh.
and we tried to get, we chose a scaling to try and get that right, which is a decision, but it's not crazy. So I think, again, I'll repeat it will be important when you get results from this to test against what other people see using different methods. Okay, so I just want to start with acknowledging the person who's done most of this work. She's a joint PhD student of mine and Jonathan Martinez, so we're jointly supervising this work. This is Marie Forrest, and I didn't have a picture of Marie, so I chose a picture of a forest to show you instead, and I seem to have not got rid of this quote, which I was tempted to get rid of this quote. So there's a sort of... I'm sort of British, and it's kind of embarrassingly kind of gushy. But I'll explain what it means in a second. So the basic idea is, this is obviously it's a bit of a joke, but you can think of what we're doing as kind of building a forest in some sense. Each of these trees might be a region. Actually, we build lots of trees per region, and then we kind of add information across the whole forest. And so what this quote is actually about is he's an actor and movie director, but what he really meant, I think, is to make a rather deep statement about how to, by yourself, he means how to find out your kind of demographic population genetic history. And we're about to get lost in the methods of building the forest of life. Okay. Okay. So There are various things we could do if we could build these trees, but one obvious goal is to understand population history, and it seems like a good fit for lots of people in this meeting. And of course, one reason to do that is because it's so fascinating and interesting and a deeply important question. Another reason to do it, from our point of view, we sort of forced to do it because we want to build trees that are realistic, in the case of humans and potentially other species in the future. And we know that these things affect the tree shapes very strongly. And so we're sort of required to try and fit a realistic model, even if we just think of it as a description of how the trees actually look. Okay, so in that kind of descriptive world, you might think of some of the interesting phenomena that you'd like to separate, like substructure or large population sizes, which would lead to similar signals, like here, as leading to rather similar tree shapes, at least in a broad sense. Okay, so we've concentrated at first on this question, and of course many of you have also worked on this in different people as well. And in general, there's been a real advance in the last few years in learning about human population sizes, in particular with the advent, due to computational innovations, of these epoch models, which assume a piecewise constant. You could fix or vary the epochs, but basically you assume a piecewise constant population size, and you have no further assumptions, so you can fit a very flexible history. And this was, first really done at a large scale by Leon Durbin, who developed a method of analysing a single entire genome to learn the effective population size backward through time. And more recently, there have been other authors trying to implement extensions of this approach, published in some unpublished work. And so it might be worth sort of pointing out some strengths and weaknesses of these, or this approach in particular. So it seems to be very powerful at learning population sizes in kind of this time range, 20,000 to a couple of million years. It's more uncertain at large timescales because there are fewer coalescence events left, but also at small timescales it's uncertain because with a sample size of 2 you don't see enough coalescence events. And so you might hope that in a sort of tree-based setting you could have a larger sample size and do better at this end of things. Another property of these methods is that they don't always incorporate splits between populations directly. So when you see populations separating, because there's some natural smoothing, as you can sort of visually see, they might start to separate before the actual split time. So it's not straightforward to go from this to sort of learn, for example, when all of these non-African populations down here and the African population are separated from one another. Okay, all right. But in general, kind of building on this work, we're trying to use a similar framework with an epoch model of population size changes, but we sort of extending this by also allowing populations to separate at explicit times in the past, and so we can get different population sizes in different populations. Okay. Don't know why that's looking so grotty, and I just do this. So, as I said, there's been a lot of previous work, and that's told us something about these split times. So mostly these models don't allow for variation in population size in that very general way I showed on the last slide. But nevertheless, in the last few years, there have been lots of different estimates on large-scale data sets of things, including the divergence time between Africans and non-Africans. And by the way, this is a reproduced figure from Scally and Durbin. And split times between Europeans and East Asians. They're in blue and green, respectively. And what you can see is there's a kind of, sort of a consensus between these methods, but they span very wide time ranges. From the point of view of what I'm going to show in a minute, that's good because it gives us a decent sporting chance of at least agreeing with somebody. But little chance of agreeing with everybody. And especially in the case, for example, of the East Asia split, although it's clearly earlier than the out of Africa split. And so there's some difficulty in the field from this. There's also difficulty, as Molly sort of beautifully pointed out in her talk, with the fact, and this is what the open and closed boxes show us, that we don't know the mutation rate within a factor of 2. And so these sort of numbers can be doubled or halved as you see fit. And Scally and Durbin argued for the slower mutation rates corresponding to the older times for a variety of reasons in their paper, but one of which was a kind of sense that there should be some correspondence, though not necessarily exact, between anthropological evidence for sort of when humans got to different places, although it may not have been the first or the last time they got to those places, and these split times, and there's something of a better fit when you use a slower mutation rate. And so we've gone with that, and we've assumed through all of this, we've got a mutation rate of 1.25 times 10 to the minus 8 per base per generation, and a generation time of 28 years, not too far off what Molly suggested tentatively yesterday of 30 years. And this corresponds roughly to the kind of direct pedigree-based estimates of human mutation rate. And so one comment about this, this would mean that we're able to look back about 2 or 3 million years into the past. So the average tree depth is going to be roughly sort of double the pairwise coalescence time. Okay, so we can look a long, long way into the past in principle. All right. So now I'm going to impose, so this is where we get lost in the forest. I'm going to impose on you a few method slides. I'll try not to spend too long on them. And so our framework, as I've said, is we've got this sort of split model, but within that we've got fixed epochs, and the population can vary in size. And we're going to assume that, say this is Africa and this is Europe, that they evolve independently post-split, so we haven't yet incorporated migration into the model. And for the next few slides, you might want to remember that we have these parameters, the population size in different epochs, call them NK, and we'll call the full set of parameters, including the split time, some vector lambda. So what we'd like to know to fit the model is what lambda is. And so what's the effect of these population sizes on the underlying coalescent tree? That's what's shown up here. Well, we're just using the standard theory, which says that when the population size is NK, chromosomes, then the pairwise coalescence rate between lineages is inversely proportional to that size. So when the population is big, like here, there isn't much coalescence. And when it's smaller, like here, coalescence happens more rapidly. And this is how we can relate the shapes of the tree to these sizes. And what we need to concern ourselves with in building trees is the sort of unseen tree, including the coalescence times and the mutation events, which aren't shown in this cartoon. And we know one thing, which is why trees are so powerful. If we can work out, if we know the tree, it's easy to write down its probability given some set of parameters. Okay, and that's because backward in time, the tree evolves as a Markov process, and so we can just write down the product of each probability of different kinds of events. The tricky thing is actually getting the tree in the first place. And so what do we actually do? So I'll talk quickly about how we build a tree given some diversity data. And so our first step is that we have some guess, if you like, at the population sizes and the split times through time. So here's such a guess. Given that guess, we try and build trees given variation patterns and given the sizes. And then we combine information across all the thousands of regions we've got and use that to update our beliefs about the population sizes and the split time. And we iterate, attempting to find the maximum likelihood history. That's the basic idea. And we have a lot of computational work to do in implementing this idea in practice. Okay. So I don't want to give you all the details because we might be here a while, but sort of an important thing is the basic idea of how one can go from a data set like this is the same toy I showed at the beginning, to a tree. And our start point for this work, it's not quite where we end up, is a sampler, what's called an importance sampling approach, and we use a method introduced by Stevens and Donnelly back in 2001. And this is a deceptively simple but powerful way of going from this data set here to sampling a random history that would produce the data that you see. Okay, so what we want to do in general is to sample the history given the data. And what one can do is sort of stare at it for a second and think about what could have happened next in the past. And there are different things that could happen next in the past, coalescence events or mutations. And if you think about it a little bit more, only the black mutations that are seen in only one sample member could be the first to mutate as we look backward in time. If they're seen in two individuals, they'd have to coalesce first. So these events could have happened, and only identical sequences can coalesce back in time. So these guys, B and D, could coalesce backward in time. And this sequence can't be the next to have an event back in time. So 5 of the 6 sequences potentially have an event. And the Stevens and Donnelly sampler says do the following. Choose one of those 5 possible sequences at random and perform the event you're supposed to. Okay, so in this case, we choose this sequence and then it has to coalesce with sequence B, and then we end up with it putting this event in the tree. And then we sort of iterate. So after that, there's one fewer sequence, if you like, because of a coalescent, and we have a new data set, and we just go around and do the same thing again. And what you can see is that after a few steps, you'll end up having built a history that will produce the data that you've seen, and it does that in a random way. Okay, this isn't the real way. This isn't the proper conditional distribution of this given the data. It's an approximation, but it turns out to be the best known approximation to that process. Right. Okay, so that's a scheme that's been used by various authors. It hasn't been used really in this split model, and certainly not in a variable population size model. Although Rasmus, back in 1998, adapted a similar scheme from Griffiths and Tavre. It wasn't quite as efficient into a similar setting. And so, how do we try and use this idea in our world? Well, it's sort of straightforward in a way to extend it. The first thing we have to do is actually add times rather than just choosing what type of event. And so we do that according to the model. And the second obvious thing we have to do is we can't have this fella here and this fella here coalescing if they're in different populations. So we ban those events from consideration. But otherwise, we can just use this approach. And that's what we did. That was the talk I'm not going to show you because that behaves very poorly in our setting. Okay, so in what way did it behave poorly? It tended to flatten out all the interesting stuff. So you'd see a history that would look much more like a constant population size than it should. And if you know your importance sampling theory, that might seem slightly, well, you probably wouldn't be so surprised at this, but the importance sampling theory says that it shouldn't really matter which choice of sampler you use if, being a statistician, you take infinitely many histories out of that sampler. So as statisticians, we like to do things infinitely many times. But actually, in practice, it doesn't work because it never simulates histories that are realistic given the data. And that's because it's designed to deal with a constant size population case. So it's not surprising it sort of pushes you towards that answer. And to solve this problem, after lots of attempts, we came up with a following idea, which is pretty simple, which is that we had the idea that we have to choose among the events differently depending on the history. And so here's the basic idea. I don't think I'll go through lots of detail on this slide. Suppose that suppose we only have one population for now, and it changes size through time like this. The idea is that we introduce a term beta j, or a vector that varies by epoch, that kind of upweights choosing the mutation events relative to the coalescence events. And so the idea is when the population size is big, coalescence happens rarely, so you should upweight your possible mutation events. And when the population size is small, you should downweight them because a lot of coalescence should be happening. And so this is the basic principle. And in practice, we choose bj to satisfy the following equation, which you don't have to understand well. But the basic idea is we're trying to force the average across trees of a mutation or coalescent probabilities to be the right thing. So we sort of end up with a sampler that's on average doing the correct thing. And we produce that sampler by simulation. Okay? And that's allowed because it's sort of formally allowed because, as I say, any sampler, in principle, if you do enough trees, will work. Okay, so we have this modification that basically makes the trees reflect the history better. All right. So the next thing that we have to do, given that we use this to build a tree given a history, and the next question is then, once we have these trees, how can we sort of estimate the historical parameters from them? And a very natural thing that we could try and use to do that, we're operating in a big parameter space. And what we think of having is we've got our variation data D, and that's sort of partial data that tells us something about a full data set that we wish we had, which is the tree, the whole history. And because H implies D, we can think of our observed data as a subset of this full data. And in that setting, we've got a big parameter space. We want to maximize the likelihood. We've got incomplete data and we think of complete data. The usual thing we think to do as statisticians, again, is to use an EM algorithm. Okay, an EM algorithm is a beautiful thing that's sort of guaranteed to improve the likelihood stepwise, so we can choose a better set of parameters given a current set. And what we're told to do in that procedure is to maximize over possible lambda something called a Q function. So lots of you will know a lot about this, but if you don't, there's this function, which in our case means that we have to sum over regions J some function of the tree that depends on the parameters we're looking for, and that function we have to take the average of over possible histories given the current parameter set. And this is why other people aren't doing this. It's because this is actually a very hard thing to do. And so all you need to really take from this slide is that to do this properly, we need to be able to integrate over all the possible histories at all the loci. And if we could do that, we wouldn't have to do this in the first place. Okay, so we can't actually fully apply this because we need to sum over infinitely many histories. And so we use this MCEM, where MC stands for Monte Carlo. And the idea there is that instead of summing over infinitely many possible histories, we sum over finitely many possible histories to estimate the Q function. And so what's the effect of doing this? The effect of doing this is that there's some stochasticity in the procedure, so the new parameters that we choose won't always be a better fit to the data than the ones we had before. But if we built a lot of trees, the theory again says, again, if we have an infinite number of trees, this will do exactly the correct thing and get us the right EM algorithm. Okay, so one problem with that is we haven't got the computational resources to do it infinitely often, but it turns out that in practice, after doing lots of simulation studies, we can get away with a rather small number of trees per region. In fact, we often only use 20 and 200 for certain steps in the algorithm. So in the results I'm going to show you, what we've done is we've taken our 2,500 regions, and for each of them, at each iteration, we've built 20 trees, and that allows us to do lots and lots of update steps to hopefully really fit these parameters.
You can't reuse the simulations from a previous update by changing the importance handling weights? So you could just start with one set of, that wouldn't be efficient? Or at least you could get more information out of keeping those histories?
Yeah, we don't do it. That sounds a bit like a kind of bridge sampling approach almost, doesn't it? And I think it's
keep all your old trees, that's it, and then simulate some new ones. Let's say you're in a part of the space where the old trees are not supported very strongly, then you would need to do some more simulations.
There's one
you should only do better with keeping the old trees. Right?
Yeah, I mean, there's a technical issue here, which is when you change the split time, lots of the trees become impossible. So it's hard to do that across split time.
But then you just have an importance handling weight of 0.
Yeah, and what we do in practice is we just deal with different split times independently. Okay. And I think it is possible, but once you're close to the maximum, you could try and do this. Although I'd have to, you'd only have to kind of change the importance weights, wouldn't you? I think it makes sense.
Yeah, but the problems we did, which
it's
are
a clever idea, and we don't
ones,
do it.
And the ones that we did that they're rather different, the sort of pedigree analysis ones, we never found it worked. It was never efficient to try and save the old ones and reweight them.
Yeah, I mean,
we tried it, done many problems. And
it's a question of how different things get,
exactly.
And it might have subtle answers, right? I'm guessing, but the trees are very random, so I think it's worth thinking about. Okay, we sort of found it worked pretty well without that cleverness. Okay, so I'm going to show. It's sort of important in this setting to understand, how we behave in simulations, and it will turn out to be instructive for the real results. So here's a simulated history. We've got it's very, very boring. We've got two populations. So here's the time in the past. It's on a log scale, so this is 10,000, 100,000, and so on years. And the population sizes change through time, and they split at this time point here. That's what this means. Okay, so we've got one that stays at 10,000, the other one increases to 20,000 at the split. And when we run the inference, this is what we get, and it's a pretty good fit. Okay, so it's not a difficult problem, but a pretty good fit. But I want to point out one thing to you, which we see in nearly all the simulations, which is that we still see this kind of smoothing of sudden jumps. And this is a bit of a surprise because we have no prior that these things should be smooth, and so it's not a result we completely understand, but I could discuss ideas for what's going on with people who are interested. You might think we'd kind of get stuff that was wrong, but kind of in random directions. I think it's also true that this history is at least as or more likely than the truth under our model, so it may be that there's very little information in the data to separate this sort of sharp jump from a smoother one. Okay.
Do you tie one of your epoch population size changes to the split time?
We have the epoch has to end at the split, right?
Yeah.
That's all that we do. So they're
On
not.
either side of the split, you have different population sizes.
Yes. So there's no. Yeah, it doesn't. It's not obvious why it smooths in this way, but it always does. So we can get some confidence intervals on our estimates, although they don't seem to well account for the smoothing. But they do show you at least that you're more uncertain as you go further back in time. And I think it's kind of important. One thing that is important to check, the most important thing perhaps, is what happens if we're wrong about the idea that there's no recombination. And so we simulated recombination at the absolute upper bound of what we think these regions could have on average. And you can see that at least for the recent past, it has almost no effect. For the distant past, it seems to have little effect, and there's this sort of period in the middle where we slightly underestimate the population size. That's probably because we're removing sites that fail the 4-gamete test from this part of the history, is my guess. But it's not too serious. You wouldn't be badly misled, you might hope, in that setting. And we also simulated a few other things. So we didn't see too much effect if we had a bit of admixture, so that there was an admixture event at about this time from this population to this one. And actually, sometimes the underestimation is less than you see here, so there's a bit of an effect. But the estimated split time is within the range of the real split times, if you like. Or if we have a more continuous migration scenario, and if I used other sort of tested possible histories, we get similar robustness effects. And we also seem reasonably robust to between 0 and 10% admixture from a sort of, let's imagine this as Neanderthal, into, say, Europe and not Africa, in terms of estimating the relationship between, say, Africa and Europe. Okay. Thank you. Okay. So this shows you that as the admixture fraction increases, for different real split times, you sort of retain surprising accuracy about the split. And some other methods, so the reason we did this, so this is from Alvarez and et al., and they found that sort of if you use the site frequency spectrum, there could be some sensitivity to this sort of phenomenon.
Does that not worry you with 10% admixture? It doesn't change the estimates? I mean, it should change
No,
the
I
estimates.
think I understand it, at least. So it's because you're kind of estimating, in some sense, the minimum divergence time between this and this, so that's not affected by this admixture event. So, I mean, some things might be sensitive, but not that time estimate, I think. All right. So here's another simulated history, and I'll go through this one quickly. Only thing to say, it's a bit more exciting, has a bottleneck, and some size changes in the past. And we see largely the same thing. We're approximating the truth, but we tend to over-smooth the sort of suddenness of this population size jump. Another thing that we often see in general is that we do reasonably well at times close to the split, perhaps because we get unambiguous information about these mutations being earlier. And so, we really nail the bottleneck at first, but then we kind of smooth it out as we're further away from that hard information from the split. So you might think that using pairs of populations with different divergence times could be informative for that reason. And then here's another history with a really large ancestral population, and we seem to do particularly well in this case.
So, Simon, are you saying that when you have two populations that both descend from a split,
Yeah.
you have much more purchase on the split time than you do if you have only one population?
Mm-hmm.
On the population size change that occurs at the split time?
Yeah, at the split time, yeah, right.
So
You get
that
resolution to
second
see what happens right then?
allows you to detect these discontinuities.
Yeah, I think so. I mean, you can sort of see why, I think. Okay. All right, and so I'll rush on to what happens when we apply this to real data sets. And we're going to assume now that those simulations make you feel completely happy about the method. So a selection of 1,000 genomes population. So the first thing I want to show you is what happens when we ask about the history of the British, self-interested, and the Yorubans. And this is what we get. So the red estimate here is the split, and this is the estimate of the British, or really the ancestor of the British, the sort of Africa population back here through time, and it's an estimate of the size of the Yoruban population through time. And you see something that's at least interesting. So the first thing we do, we have an estimated split time of 80,000 years, rather 79,739 years. And at the split, encouragingly, given what we know about human history, there's a sharp bottleneck, and Europeans nothing whatsoever in the Africans. And predating that population split, this is when we have this large population size, and that might reflect, people have been talking about these ghost populations. It could easily be ancestral structure, or it could be a genuinely large size, and then further back, an interesting trough. And so one noticeable thing is that bottleneck is completely specific to the out of Africa group. There's nothing at all happening in Yoruba. In fact, they steadily kind of seem to decline in population size since. And what we can ask is so firstly, this is the likelihood surface, which kind of looks fairly flat at the top, but if you zoom in a bit, the likelihood differences are extremely large. So that formally, given these confidence intervals in our likelihood estimation, you can exclude these times, all these other times for the split, and you get sort of a maximum confidence interval of this sort of size. And I think relatively interesting thing, when we apply it independently to other pairs of populations, so they always involve YRI, we get almost precisely the same split times with independent data, at least on this half, and are always overlapping confidence intervals. And so, and here's the population size estimate. I haven't just shown you I feel a bit like I might have accidentally reproduced the same plot 4 times, but you can see that there's extremely concordant estimates of history, and here's the uncertainty. So we feel like we're reasonably confident even a longish way into the past. And so if you sort of lay them on top of one another, so here's the British African one that I showed, and here's the African Han Chinese, and it's pretty identical. Same split time, a little bit of difference here, stronger bottleneck apparently in the Chinese group, but otherwise they're really incredibly similar. And one thing that we might also do is ask how this compares to the previous work, right? So we can compare to Lee and Durbin's. So they don't explicitly have the split, so we might expect things to differ a bit around here. And we also think that we have a bit more information from having more sequences at these earlier times, so things stay reasonably well behaved and stable. But you can see that at the older times, at least, it seems like a pretty good match to me. It's not quite clear if they exactly line up, but given the uncertainties and everything, they're pretty similar to one another. So we're encouraged that there's nothing going terribly wrong here. And so one thing that you might then do is, as I say, sort of try exploring other pairs of populations to see if you can learn more about what's happened, say, since the Out of Africa event. And the idea being, based on the simulations, we see this sort of steady increase in the UK ancestors or the Chinese ancestors, but we know that we'd expect that to be smooth, even if there were, say, a long bottleneck and then a sharp increase or something else. So this isn't probably very informative about the details, so we could hope to use different splits to gain information. And when we do that, for different Europe-East Asia splits, we get estimated split times which vary a bit more, interestingly. So they're from like 42,000 to 52,000 years. They're older when we have Japanese than when we have Chinese. I don't know. Our confidence intervals would suggest this is a significant difference. But again, we see pretty concordant histories, and this tells us at least that it doesn't look like the population size has grown appreciably by the time, China and the sort of East Asian and the European population split. So it's still small. That's probably half the time from this Out of Africa event to here. The population size hasn't grown appreciably. And the other thing that's encouraging is we can still see the Out of Africa bottleneck even when we do that comparison. So my time's up and I'll go faster and faster. Okay, but I do want to show you a few more things. So sorry, Ian. So one thing that we might then ask, an extreme case of what we might try and do with this method is take two very closely related populations, in this case the Tuscans and the British. And I think the results are interesting, but they're uncertain. So the first thing we found is that we had trouble estimating the split time between the UK and Tuscany, which is in northern Italy, but we can tell that it's not 0. So they have a split between 1,000 and 6,000 years. That's about what we what we estimate. But I think the interesting thing about this is no matter what time we fit, we find that the vast majority of that expansion that we've seen, that was smoothed in the earlier plots, seems to actually post-date the split of those populations. So you see that they're remaining seemingly pretty small throughout the Ice Age, and it's almost like they grew in size, on their own yesterday. That's sort of what it looks like. And there's this, we always see this little bump that's a bit in, again, it could be structure of some kind, at about the sort of 10 or 20,000 year point in the past. So it's a little bump, but it's pretty resilient. And again, we get consistent dates for the bottleneck, but we're smoothing this population size a bit further into the past. Okay, so one caveat to this should clearly be higher, because we know that 2/3 of the singletons have been missed in 1,000 genomes. So the main effect of that will be that these population sizes will be probably somewhat underestimated. Okay, so just to say, I told you we'd agree with someone. So we're at the bottom end actually, but we just about agree with Simon Gravel, and we're in better. We nearly managed to not agree with anyone in some of the European Asian splits, but we just about get in there, and the fossil dates are not so terrible either, but it's hard to interpret. And the final thing I'll show you, this is something we shouldn't show you. So what happens if we use Neanderthal? Well, we're pushing our luck because it's a long way in the past, so maybe recombination's got a lot of time to happen, and also we don't actually know that the PRDM9 allele carried by Neanderthals is the same as humans, so they could even have a different recombination rate to modern humans, and so could the ancestral group. We just went ahead and did it anyway. And we know from the recent Altai Neanderthal paper, we've got a split time estimate of 550 to 765 years, or in our scaling it's this. And so when we did this, here's what we got, 630, 640,000 years. And we suspect that this is not evidence of no inbreeding with Neanderthals. We don't think that will much impact the split time. We actually see a strange distribution here where there's a bimodality of the split time distribution whenever we use an out-of-African group. And when we use the African group, we also see something a bit strange, which is that these two things are sort of almost equally likely. So the split time could be up to 1 million years, at least according to our method. And this seems to be mediated by the fact that this is a large population size, so we don't see many coalescences in it. And so we're pretty uncertain from the African groups about that split time. The last thing I'll say, I promise, is that one thing we really see is there's, again, a very strong instantaneous bottleneck in Neanderthal at the split. And so, I'm sure that you guys have found, right, that they have a really small population size, and PSMC has seen this. But it looks like, it happens instantaneously at the separation, as if maybe they were the group that left. And it stays small for a very long time. So this is why there's so much less diverse in humans. It's staying small for a really long time. And even in this setting, we're doing okay in terms of when the bottleneck happens in the Europeans. So you might think, just to conclude, we're not doing so badly, I think, over a very wide time range, a few thousand to a few 100,000 years. And I'm out of time, so just say, we're hoping to do more complicated realistic models. And those include kind of directly simulating the trees better, but also trying to interrogate the trees to see if we see funny patterns based on what we expect. Okay, and I'll just finish by saying, so Jonathan asked me to point out that we're recruiting. So if you're interested in postdoctoral positions, either in Jonathan's or in my lab, we're recruiting at the moment, and also within the stats department, there are more senior positions available if any of you are at that stage. And I just want to thank the people involved in this work. Anna did the Neanderthal stuff, and lots of you for helpful discussions. Thanks a lot.
One quick question.
Yep.
I guess I was wondering how informative the data is about individual trees. And could you actually spot the split time just by simulating trees under a no-split model and just looking for sort of deep coalescence between, say, Africa and Europe?
I think that would have. So they're pretty informative, given they have lots of mutations. I think that would be biased based on the poor results of a vanilla sampler, which tried to use a constant size population in some sense. So they're not quite informative enough to avoid you getting those answers wrong. I guess the issue is that some of the details are influenced by some cases where there are large population sizes near the split. And once you notice there are large population sizes near the split compared to later, that pushes your split time forward a bit. And I think those sorts of subtleties don't come out unless you try harder.